Hi students, good afternoon. Welcome to online classes. I am U Bhargavi CRT Biology from KGBV Kalandu. Today we will discuss about cell, its structure and functions. Before going to this lesson, in the previous class we know some information about cell. Cell is a basic unit of all living things. You know, each and every organism made up of a cell. It is similar to our wall. Wall are made up of a bricks. It comparison only for understanding purpose. Cells are observed under the microscope. I mean, we cannot see with our naked eye. Cell contain cell wall, cell membrane, cytoplasm, nucleus, chloroplast and mitochondria, etc. Cell discovered by Robert Hooke and living cells are discovered by Antoine Leeuwenhoek. Robert Hooke discovered cell in oak stem. This is dead cells. But Leeuwenhoek discovered living cells as sperm cell and RBC, etc. The study of cell is called cell biology. Dear students, I give you some questions. Please write down your notebook and answer them. The first one is what is cell biology, and another one is who discovered living cells. Discuss about typical cell. Typical cell is a model cell. The typical cell provides a way to study cells. All the organelles shown in the typical plant or animal cell will not exist in every cell. Example, chloroplast is always in the typical plant cell, yet all the plant cells do not have chloroplast. They are mostly found in the green leaves, tender stems of cell. The organelles that features in the most of the cells are included in this model cell. I mean, it is a model for understanding cell structure. It is only study purpose. See these slides for typical plant cell and animal cells. Yes, children, some more questions. First one is write down the differences between plant cell and animal cell. Draw the diagram of typical plant and animal cells in your notebook. Cells. Parts of cells as below. Cell membrane. Cell membrane is known as plasma membrane. Cell wall, nucleus, cytoplasm, cell organelles. These are the major parts of the cell. In this, cell organelles are mitochondria, plastids, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes and vacuole. The major cell organelles are in the cytoplasm. Now we learn each and every part structure and function. Now I will discuss cell membrane. Cell membrane is also known as plasma membrane. Plasma membrane present in both animal and plant cells. The outer boundary of cell is called cell membrane. What is meant by cell membrane? The outer boundary of cell is called cell membrane. Cell membrane is flexible and is made up of proteins and lipids. It is separates cytoplasm from the external environment. It defines the shape and size of the cell. It protects internal parts from the external environment. Students, plasma is compared to our school compound wall for understanding purpose only. Our school compound wall protects us from outer environment. It plays a role in maintaining the balance of various substances. I mean, substances like water, minerals are maintaining the balance by the cell membrane. Any substances entering or leaving the cell only through the plasma. It is also similar. Our school gate is only enter or exit of school. This is only understand purpose. Activity. How to observe cell membrane? Please write down this activity in your lab record. We conduct the activity to observing the cell membrane. What is aim to observe the cell membrane in REOLIF? What are the requirements for this activity? Apparatus. REOLIF, microscope, watch glass, slide, needle and salt solution. These are the requirement apparatus in this activity. Before going to conduct any experiment, we have to know precautions, because without knowing the precautions, it leads to damages. Precautions. Scrapped material should be spread uniformly on the slide. Procedure. Take to it REOLIF, tear it and put it on slide. Now put 1 to 2 drops of dilute salt water on the membrane and leave it for 5 to 10 minutes. Then observe it under the microscope. Observation. Shrinking of cytoplasm along with cell membrane, which become separated from the cell wall. Result. To observe the cell membrane of REOLIF. Students, I hope you are understanding it. Let us discuss about cell wall. Plant cell contain an extra layer outside the plasma is called cell wall. What is cell wall? Plant cell contain an extra layer outside the plasma is called cell wall. Cell wall is present only in plant cells, but animal cells not having cell wall. This is the major differences between plant cell and animal cells. Cell wall is made up of cellulose. It is a carbohydrate. It is a through but flexible force layer. This is cell wall structure and cell wall function. It gives shape and protection to the cell. You know, cells are in different shapes like round, rectangular, stars, etc. These shapes are maintained by cell wall. It is continuously exchange information with other cells during growth and development. This information exchange through the plasma desmata. Plasma desmatas are connective links of cell and also exchange information. It protects outwards pressure excreted by the cell shaft. I mean external pressure towards plant body leads to damage the cell, but cell wall protect that external pressure. So plants can withstand much greater exchange of surroundings than the animals. Next activity, nucleus. How to observe nucleus? Please write down this activity in your lab record. We conduct the activity to observing the nucleus in cheek cells. What is aim? To observe the nucleus in cheek cells. What are the requirements for this activity? Apparatus. Toothpick, glass slide, cover slip, watch glass, needle, blotting paper, 1% methylene blue, normal saline, glycerin and microscope. Precautions. Do not scrap the cheek too hard. Scrap material should be spread uniformly on the slide. Excess stain should be drained off. Next, there should be no air bubbles under the cover slip. Next, procedure. Wash your mouth and scrape the internal lining of your cheek inside your mouth with a toothpick. Place this in a watch glass with normal saline. Next, then place the material on a glass slide and put a drop of methylene blue. Next, wipe off the extra strain with blotting paper. Put a drop of glycerin over it and place a cover slip. Tap to cover slip with a needle so as to spread the cell. Observe under the microscope. How to observation in this activity? Observation. We find dark colored dot in cells. This is the nucleus. Result. To observe the nucleus in cheek cells. Students, I hope you understood it. Nucleus. Now we will discuss about nucleus structure and functions. All cells contain nucleus except eukaryotic cells. All cells means plant and animal cells. These are eukaryotic cells. Prokaryotic cells are bacteria, blue-green algae. These are do not having nucleus. It was discovered by Robert Brown in the year 1831. Who discovered by the nucleus? Robert Brown discovered by the nucleus. Which year? 1831. This is an important organelle of the cell. This is also known as cell's control room. The nucleus is the largest and most distant of all organelles. I mean, nucleus can see under compound microscope also. Sleden thought that new cells were created from the nucleus and he called it as cytoblast. It included in the cell theory. Almost all eukaryotic cells have a nucleus except red blood cells in some mammals and plant phloem sieve tubes. Even these cells do have nuclei in the beginning, later destroyed while growing. This is the nucleus structure. Next, nucleus function. It is regulates and controls all the functions of the cell and denotes the characteristics of the organism. It consists of all genetic information. It is also involved in the cell division. It encloses nuclear membrane. It separates nucleoplasm and cytoplasm. Nucleus contains genetic material. This DNA controls all activities in the cell body. Nuclear membrane similar to cell membrane. Yes, children, there is dot-like structure in this figure. It is called nucleus. Can you see it? Differences between prokaryotic and eukaryotic cells. Eukaryotic cells and prokaryotic cells. Eukaryotic cells contain a membrane-bound nucleus, but prokaryotic cells cannot contain a membrane-bound nucleus. Eukaryotic cells contain all organelles, but prokaryotic cells contain only cytoplasm, DNA and ribosomes. Eukaryotic cells contain 80s ribosomes. Example, all plants and all animal cells. But prokaryotic cells contain 70s ribosomes. Example, cyanobacteria, blue-green algae. Cytoplasm. Cytoplasm is a fluid inside the cell. A large region of each cell enclosed by the cell membrane is called the cytoplasm. This fluid bounded by the plasma membrane. It contains cell organelles like mitochondria, chloroplast, etc. Cell organelles are floating in the cytoplasm. It is similar to watermelon. In watermelon, red colored pulp resemble with cytoplasm and seeds are resemble with organelles. For a long time, it was believed that the essence of life, hence this was named protoplasm. But the material inside and outside the nuclear membrane was differentiated after the discovery of nucleus. Hence, protoplasm was renamed as cytoplasm. The fluid inside the nucleus is known as nuclear fluid. Cell organelles. Structure of mitochondria. Mitochondria is discovered by Benda. It is small, spherical or cylindrical organelles. Mitochondria are sac-like structure present in the cytoplasm of the cell. It is 2 to 8 microns long and about 0.5 microns width. It is about 150 times smaller than the nucleus. It is about 150 times smaller than the nucleus. There are about 100 to 150 mitochondria in each cell. Mitochondria have double membrane. First one is inner membrane. Inner membrane is called matrix. Second, outer membrane. The inner membrane is thrown into several floors called cristae. The cristae extended into the matrix. The space between the floors is continuous with the outer membrane. This is the mitochondria structure. Next, mitochondria function. It is responsible for cellular respiration. It leads to drives energy because mitochondria is known as powerhouses of the cell or energy currents of the cell. Students, see this slide. This is mitochondria. Mitochondria have two membranes. This is outer membrane. This is inner membrane. Inner membrane is called matrix. Spherical structures like DNA. DNA means deoxyribonucleic acid. Green dot-like structures is called ribosomes. This is cristae. Activity 2. Observing mitochondria. Aim. To observe the mitochondria in onion peel. Apparatus. Onion peel, blade, watch glass, slide, cover slip, brush, needle, microscope, Janus green bee. Precautions. Onion peel should be spread uniformly on the slide. Next, excess stain should be drained off. Next, to avoid air bubbles on the slide. Next, procedure. Make a solution of Janus green bee in a beaker. Put the onion peel in this solution and keep it for 30 minutes. Keep a piece of onion peel on the slide and wash thoroughly with water. Then keep cover slip and observe under the microscope. Observation. We have observed green oval grains in the cytoplasm. These are the mitochondria. Result. To observe that mitochondria in onion peel. Plastids are mainly of three types: chromoplast, chloroplast, leucoplast, chromoplast. They are appear in different colors as red, orange, yellow, etc. Next, second one, chloroplast. It's appear in green in color. Last one, leucoplast. It is colorless chloroplast. Next, structure of chloroplast. These are of different shapes, disc, oval, etc. In algae can be found as ladders, star, spirals or reticulate. Next, the size of chloroplast is between 4 to 10 micron. It contains chlorophyll pigments. Chloroplast function. Chloroplast is to trap energy from the sun and transforms its chemical energy and it leads to carry out photosynthesis. Please student observe this figure. Activity 3. Observing plastids. Aim. Observation of chloroplast in rheoloef apparatus. Rheoloef, slide, cover slip, microscope, etc. Procedure. Take the rheoloef and mount it in water and a slide. Observe it under the microscope. Observation. We have observed a small green granules. These are identified as chloroplast. Result. To observe that chloroplast in rheoloef. Next, ribosomes. Ribosomes are small green-like structure in the cytoplasm. They are called ribosomes. These are made up of RNA and protein. RNA means ribonucleic acid. Ribosomes are two types. First one, free ribosomes. Second one, attached ribosomes. First one, free ribosomes. Free ribosomes are scattered in cytoplasm. Next, attached ribosomes. Attached ribosomes are on the surface of rough endoplasmic reticulum. Next, function. Protein and lipid synthesis. Ribosomes are two types. 80S ribosomes and 70S ribosomes. Eukaryotic cells contain 80S ribosomes. Prokaryotic cells contain 70S ribosomes. Students, see this slide. This is ribosome diagram and next one, protein synthesis process here. In this figure, there is endoplasmic reticulum. Can you see it? This is rough endoplasmic reticulum. Their surface have ribosomes. This is smooth endoplasmic reticulum. These do not have ribosomes. Endoplasmic reticulum. When the cell was observed under the electron microscope, a network of membrane was observed throughout the cytoplasm. This is known as endoplasmic reticulum. The endoplasmic reticulum is a large network of membrane-bound tubes and sheets. This is too similar to the plasma membrane. Endoplasmic reticulums are two types. First one, rough endoplasmic reticulum and second one, smooth endoplasmic reticulum. Rough endoplasmic reticulum, their surface have ribosomes. It prepares proteins. Next, smooth endoplasmic reticulum. These do not have ribosomes. It prepares lipids. The manufactured proteins and lipids are then sent to various places in the cell depending on the need. Some of these proteins and lipids help in building the cell membrane. Endoplasmic reticulum is too serve as channel for the transport of materials between various regions of the cytoplasm or between the cytoplasm and nucleus. It also functions as a cytoplasmic framework providing surface for the biochemical activities of the cell. In vertebrates, liver cells, smooth endoplasmic reticulum plays a crucial role in detoxifying many poisonous and drugs. Can you see it, students? This is Golgi apparatus, Golgi complex. These are observed by Camillo Golgi in the year 1898. This organelle is also made up of several membranes. These membranes create sac-like structure around which many fluid-filled vesicles abound. The number of Golgi bodies varies from cell to cell. Example, gland cells. Next, function. The proteins and other substances are altered slightly in Golgi complex. These are packed various substances before transported to other parts. It is also regenerate or repair the membrane. These are lysosomes. Can you see it, students? Lysosomes. The scientists are troubled for a long time that some enzymes present in the cell have the ability to destroy the cells but did not damage it. This doubt was solved when lysosomes were discovered in cytoplasm. It was found that they contained the destructive enzymes. These enzymes normally do not contact with the rest of the cell. The materials that need to be destroyed are transported to the lysosomes by the digestion and lysosomes are brushed. Hence, lysosomes are known as the suicidal bag of the cell. This is vacuole. Animal cells vacuoles are small in size. This is plant cell vacuole. They are large in size. Vacuole activity. What is aim? Observation of vacuoles in the cactus plant. What are the requirements for this activity? Apparatus: cactus peel, slide, watch glass, microscope, water, saffron solution, etc. Procedure: take thin cross section of stem of cactus in watch glass with water. Stain it with dill saffron solution. Observe the section under the microscope. Observation: we find the large empty space in the cell. These are vacuoles. Result: to observe vacuoles in the cactus plant. This is the vacuole activity. Structure of vacuole. These are fluid-filled sac-like structure. In animal cells, vacuoles are small in size, while in plant cells they are large. In mature plant cells, they might occupy almost entire cell space. Vacuoles maintain trigger pressure within the cell. They expel unwanted substances from the cell. Are cells flat? Usually, when cells are seen under the microscope, it appears as flat and two-dimensional. But in reality, cells have length, breadth, and thickness and three-dimensional. This 3D objects of cell is observed by slightly changes the focus while viewing plant cells on the slide and look at the cell wall. Cell theory. M.J. Sleeden, botanist, and Theodore Swan, zoologist, together formulated the cell theory since 1831. This theory did not explain as to how the new cells formed. Rudolf Virchow, 1855, first explained that the cells could be formed by only the division of the free exciting cells. This point also concluded the cell theory. Cell theory. All living organisms are composed of cells and products of cells. All cells arises from free exciting cells. All of you observe the diagrams. Dear students, the lesson is over. All of you answer the following questions.